Yeah.
Okay. So I think we'll start in just 2 minutes. It's getting 6:10, so we can start now. Yeah, so people will be joining. I think we'll be getting good number. Uh, so now I'll just give introduction about this event to everyone. So welcome everyone. So today for today's webinar, the AI automation of omics workflows. It is conducted through collaboration of the Nobel Research Sprint 2025, a Nobel-inspired interdisciplinary ideathon, and Boltzmann Labs, who are our innovation partner for Nobel Research Sprint and also the sponsors. They are biotech company which who are pioneering in AI drug discovery and biomarker discovery. So today, the experts from Boltzmann Labs, that is Mr. Harsha, Dr. Harsha, and Miss Gayatri, are going to give us a session on what are omics. Basically, in this whole 1 hour, they will cover what is omics and how AI can be helpful in uh, going through omics workflows. Everything is going to be through case-based discussion, and also they will be discussing about how Boltzmann Lab has been creating impact through their AI solutions. So yeah, so now we welcome Mr. Harsha and Ms. Gayatri for going forward with this webinar.
Pawan, could
So
you enable screen sharing for this?
yes, 1 minute. uh, I think now you can share. Yeah.
Good evening, everyone. Thank you for the organizers, especially the Nobel Research Sprint 2025 organizers. I'm Harshvardhan from Boltzmann Labs, and I thank you for the opportunity for giving us to present for the participants of Nobel Research Sprint 2025. So, to give you a small introduction about Boltzmann Labs, it was started in 2019 at Bangalore, and now currently we are located in Hyderabad. At Boltzmann Labs, we are building an AI native platform for molecular discovery, drug designing, drug discovery, biomarker design, small molecule design, and retrosynthesis by combining cutting edge generative AI with deep scientific insight. At Boltzmann Labs, we are aiming to make advanced drug discovery tools accessible to researchers, students, and various other industries. And also, our models can rapidly design novel proteins, optimize molecular structures, and also the prediction of synthesis pathways. So, we are passionate about empowering the next generation of scientists with tools that will help them experiment, innovate, and bring their ideas to life much faster, like reducing the uh, costing or the time. And also, our mission is to transform how molecular discovery is done and enable breakthroughs that can genuinely impact human health. So at Boltzmann, we are accelerating life sciences research. And now I hand over the session to Gayatri, a bioinformatics scientist at Boltzmann Labs. She'll be leading this webinar.
Thanks, Harsha, for that introduction. Can everybody hear me well and see my screen?
Yes, ma'am.
Great. Thank you. Thanks to the organizers of this Ideathon for having us here, and thanks to the organizers. And so we'll start our session today for AI in omics. Let me just quickly. Okay, so today's session will be on AI automation in omics workflows. So I'm assuming a lot of you are medical students. Can we have a show of hands to see how many of you are medical students here? I just want to know what my audience are today. Great. Can the rest of you also quickly? Right. Great. So most of you are medical students. Is anybody? You can stop raising now. Is anybody from medical adjacent fields like research, pharma, clinical, or engineering? Okay, great. So since most of you are medical oriented, let me start from there. What happened in early 80s, let's start the story there, because what you see in the title today are some very important buzzwords like AI, automation, and omics. But this story started in the 80s, where biology was more towards anatomy, more like what you people study, and then in towards the 90s it became more about measuring things, for example measuring genes, measuring proteins, and things like that. But later, what happened in the late 90s and early 2000s, that changed biology forever. A lot of things happened in the history of biology that changed biology forever, like the discovery of DNA structure and so on, the first invention of the microscope and all. But in the early 2000s, what happened, it changed biology forever. I dare say that it still has an impact on us. I'm Gayatri. I am a part of the omics team at Boltzmann's Labs, and today I'm going to talk about what happens when you try to use AI automation in omics workflows. So, can anybody tell me what happened in early 2000s that has changed biology forever? Any guesses from your end?
Yeah, I think they have mentioned Human Genome Project in the
Yes.
chat box, and
Okay, I can't see the chats. 1 second. Oh,
and others can
yes.
also mention your answers in the chat box.
Okay, like great. So yeah, we do have the Human Genome Project. Exactly in 2003, what happened is that scientists were successful in completely sequencing the entire human genome, and from then a lot of things changed. So what exactly changed after that is basically what where our story starts now. So first I'll talk about what omics is, what can we measure today, what does omics basically cover, what is multi-omics, and okay, we're generating data, but then how do we interpret this data, and why is AI getting into this? And don't worry, I'll take you through the lens of medicine and clinical diagnosis and treatment, because that is where omics is heading to now. Uh, After talking about omics, I'll talk about three modalities that AI can solve for us today. These three basically cover where the technology is currently. And finally, we'll talk about how Boltzmann is contributing to AI in healthcare. So, since you've guessed Human Genome Project correctly, can somebody also define what omics is, just to see your familiarity with it? Okay, I'm going to continue with this. Let's. Okay, so for a lot of time, what happened is cell was seen as the structural and functional unit of life, and rightly so, because a cell and its parts are important for life to have a structure. Cells together form tissues, tissues form organs, and they help in maintaining life. But what happened after the Human Genome Project is that people have started seeing the concept of a cell in different ways. Earlier, cell was just a bag of organelles and molecules, but something shifted after the first genome was sequenced. That means that cell is a combination of molecules and molecular pieces and processes. That means there are processes like transcription, translation, cellular processing, enzymatic reactions. All of these are happening within each cell. You're all anyways familiar with them. You're familiar with the process So what happens when you see the cell as a combination of molecular pieces and processes is that you start to break it down. And once you start breaking it down, you want to measure it. That is what happened. Biology shifted from description to measurements. Not just the cell, even if you're thinking about a patient, for a lot of time, a patient was just a set of symptoms. What happened later is patient is a diseased state which can actually be measured in terms of genomes, proteomes, transcriptomes, and other omics. Let me quickly define to you what omics is. The word om is Greek for a complete set of anything. So when we are trying to measure a complete set of anything or cover a complete set of anything, that is called an om. And when you add like genome, exome, epigenome, transcriptome, metabolome, and proteome. So these basically means that if you have a gene, you're just studying that gene. But when you're studying a set of genes, it's called a genome. If you're studying a set of expressed genes, then it's called an exome. If you're studying how gene regulation happens, that is called an epigenome. If you're studying at a given point what genes are expressed, then it's a transcriptome. If you're studying how the cell is feeding itself, right, how the cell is surviving, that is what waste is a cell releasing, that is called a metabolome. And if you're studying all the proteins that are expressed in a cell at any given point, is a proteome. So omics is basically a measurement of a complete set of biomolecules. That can be the genes inside a cell, that can be proteins, metabolites. So can anybody tell me what's so special about being able to measure a complete set of molecules at any given time?
People can put their answers in the chat box. Yeah.
Okay, we can map out the mutations. Any other answers? Okay, so for a lot of time, again, pinpoint individual variations. Great. And create therapeutic interventions regarding the same. Great. So again, if we go back into history, the thing with biology was that it was always we were always focusing on one point at a time. So one disease at a time, one gene at a time, one protein at a time, one mutation at a time, because we did not have the technology to look at it as a complete system. What we can do with omics right now, because at any given point, we are today able to look at the cell and exactly map out what genes are being expressed, what mutations exist at that given point in time, what proteins are present in the cell, and so on. So what does omics tell us? Omics tells us something what is called a biological state. This is a new concept even in biology, because now biological state means that at a given time point, if you are taking a cell, a tissue, or a patient, what are all the molecules that are expressed at that given time? What all pathways are active? Something like that. That is a biological state, and that's what omics helps us map and measure. The key word here is to understand that we are able to measure. Earlier, if you wanted to measure something, all you could do was count cells. I think you could use, how you count blood cells on a histopathological slide, or, that is the kind of measurements you could do. Or what we could maximum do was take a cell, pull down all the proteins using centrifugation, then Western blot it, and then we run it on a gel. But that is also semi-quantitative. You don't know how many protein molecules are expressed. You don't know how many gene molecules are expressed, and so on. And now we'll come to why you guys are trying to understand this. Why are we talking to you guys instead of maybe going to a bioinformatician or to an AI engineer, right? Why do medical students, why do clinicians have to understand this? Again, this is a huge shift in biology where diseases are usually defined by symptoms, but here, or imaging symptoms, and blood tests. But diseases alter your molecular profiles even before symptoms occur. Today, if you have to have a positive test for diabetes, your molecular profiles would have changed sometime before that. Omics measures these changes, reveals mechanisms, and helps us predict treatment response as well. And how do we do this? We do this through high throughput next generation sequencing. And high throughput next generation sequencing is basically wet lab techniques, which we use a very novel technique called next generation sequencing, and we pull down all these molecules, separate them, amplify them, and look at them. Don't worry about how this is done for now. Because what we are trying to understand today is not very in depth into how these data is done, but once you have this data, what do you do, and how is this data even relevant to clinicians like you all, future clinicians like you all? So, can anybody tell me why you, as a future medical professional, care about these things from whatever we understood till now? Because so far, if you prescribe a drug for somebody who comes in with symptoms, that seems to be enough. Why do you even want to go to all of this pain? We are moving towards targeted cellular therapies, but for what? Because see, just because something is available, it doesn't make sense to use these things, right? Because it involves a lot of cost and all. Right. I can see some good answers, precision, early diagnosis and prophylaxis for better diagnosis, also for better treatment. And I will tell you why quickly. So one thing is that if you're familiar with the clinical trial, general drug development cycle, and also the clinical trial cycle, 90% of the drug candidates fail, and 40 to 50% fail due to a lack of efficacy, which means that once a drug is tested, first preclinical, clinical, and it reaches clinical, it is failing because it's not standing up to what it was developed for, and we need better target validation. Even before it's going into a subject organism, it has to be validated with and to predict the clinical success, because a lot of cost is involved in this. Also, traditional single biomarkers may fail to capture disease complexity. So, what we use traditionally, the biomarkers for any cell, for example, TP53 gene is a good biomarker for cancer, but that has been done, that has been discovered through years and years of wet lab research. And whatever genes we have right now as biomarkers were all out of experimentation, and some of them through coincidence. Multi-omics profiling identifies which patients will respond to specific treatments. This is especially used when you're trying to test a drug. And it helps in knowing your patients well in rare diseases or in very heterogeneous diseases like cancer. And also classical methods miss cell specific disease mechanisms. You understand, you see that sometimes, especially in cancers, you see disease resistance, drug resistance, disease heterogeneity, and other mechanisms. And a drug which is tried and tested also doesn't work sometimes. And one of the most important contributions of omics and AI is the time and cost cutting ability. You can rapidly repurpose existing drug cycle. I remember that when I was studying about repurposing drugs, can you tell me a classic example of drug repurposing? There's one very classical example that they give for drug repurposing.
Weight loss
Drug-
drugs.
Sorry?
Weight loss drugs like GLP analogs,